Alrighty, good morning, everyone. I hope that folks can hear me well. Welcome to our web-based demonstration workshop. And thank you for joining us today. My name is Anya Starovoitov. I am a project manager with Sonoma Resource Conservation District. We're here today to talk about soil health, so we're excited that you can join us. I first wanted to give a thanks to the project funders. The project we're talking about today is funded through the Healthy Soils Program. And the Healthy Soils Program funding actually comes from the Greenhouse Gas Reduction Funds, which is part of the California Climate Investments statewide program that uses cap and trade dollars to do work associated with reducing greenhouse gas emissions, strengthening our economy, and also improving public health and the environment. So we thank them for moving some really cool, innovative farming practices forward with this funding. Also wanted to say thanks for funding and project support from our partners, Jackson Family Wines. They're here today with us for the presentation, and we are excited about this collaboration. And let's see, some really quick general info about the way this workshop will go. We are going to have a few presentations and we're going to just roll through them. And I invite you to ask any questions that you might have as the presentations go on using the Q&A button at the bottom of your screen. You could input a question, and then once the presentations are over, we will have a nice long time for some question and answers with all of the presenters. If you are having any technical difficulties, you're welcome to use the chat window and let us know if you're having some problems. Alrighty, and let's see, we will get rolling with Dr. Josh Beniston. And Dr. Beniston is an instructor and director of sustainable agriculture at the Santa Rosa Junior College. He previously worked as a research scientist with the U.S. Department of Agriculture's Agricultural Research Service and focused on soil health, soil carbon cycling, and managing soil for urban agriculture. He has a bachelor's in plant biology, as well as a master's in soil science and a PhD in ecosystem science from The Ohio State University. And I will let Josh take it away to tell us more about what our joint project is, and then some of the exciting soil health data highlights that we have from this year.
Okay, good morning, everybody. Thank you for joining us, and thank you for the introduction and the invitation, Anya. And so, yeah, what I'm going to do with my presentation this morning is just give you a little bit of background and introduction to our project and what we've been working on. And then I'm going to show some results from our 2020 soil data and soil sampling. And so this project, as Anya mentioned, is a collaboration between a lot of different groups. It's hosted with the Jackson Family Wines and Sonoma RCD, and Anya were able to get the Healthy Soils grant for us. And then myself from SRJC and other partners from University of California and USDA. And then the California Department of Food and Ag has funded and overseen this project. And so the project comes from a specific area in CDFA called the Healthy Soils Program, and a subdivision of that called the Demonstration Project Program, which really seeks to apply healthy soil practices on working farms. And then as a demonstration project, we're obliged to both collect robust soils data on our healthy soils practices, and also to share it with the agricultural community. And so this workshop today is part of our commitment to that. Our goals with this project were to assess the effect of compost applications, as well as different traditional tillage methods on soil health and soil carbon content in vineyards. So that's the focus. And major activities for the project that I've been involved with is we've been doing an annual soil sampling, and soil data collection at the site from starting in 2018 and then continuing through this year and continuing for a few more years into the future. And then we've been doing an annual field day and workshop about the project, and so that's what we're here doing this morning. So to show you what we've done, and to mention, I guess, to back up, so this is hosted at a vineyard called Saralee's Vineyard. This is a very nice vineyard right in the Russian River Valley and bottomland soils in Santa Rosa, on the northwest side of Santa Rosa, just off of River Road on Slusser Road. And so that's the location for this site for Sonoma County folks who are joining us. And so what we did at Saralee's Vineyards is that we put 3 different tillage treatments in place to compare them. So there's a full tillage where both rows on either side of the grapevines get tilled each year. There's an alternate row tillage where one side of the vines gets tilled each year, and those alternate and flip back and forth on an annual basis. And then there's a no tillage treatment where there's no tilling going on in the tractor rows between the vines. And we have a second experimental treatment in our design, which is our compost treatment. And so this is then, nested within those tillage treatments. Some plots get an annual application of 5 tons per acre of low nitrogen compost, I believe it is, and others do not. So these things are both happening. And then we've set this up in 2 vineyard blocks, and each of them has a different grape varietal. There's a Chardonnay block and a Pinot Noir block, kind of major Russian River grapes. And then we're doing soil sampling in both the tractor rows and the vine rows. And so then here's a picture of the whole experiment, right? So you can see this. If you look at these colored bars, or colored columns, I should say, each of those represents the different tillage treatments. And then there's a double, so each tillage treatment has 2 vine rows, and then both sides, both tractor rows on each of those vine rows, and then one set of vines and tractor rows is getting compost, and the other set is not. And then there's a space. There's then a full vine row between different plots. So, as an agricultural scientist, I feel really good about this design. This is a robust design. It's a large area. I was surprised when I was putting my presentation together that it's, I should say that it's only 5¼ acres. Having walked it now every spring for a few years, I would have guessed it was larger than that. But it's a nice, large, robust experiment. So we're very fortunate that Jackson Family Wines and the vineyard team here has been on board to do an experiment at this scale. It's exciting to do it at a real farming scale. And so, as I mentioned, we're collecting soil samples each spring for the project, and I'll show you some data from those from this year. So we collected those in 2018 and 2019, and then this year a couple assistants and I got out there just before Sonoma County shut down. Literally the week before, we were out there collecting soil. So it was good timing. The spring came earlier this year as it was a drier winter, and so we got out there sooner. And we're collecting what we call composite soil samples, which is essentially that we walk both of the tractor rows in each experimental unit, and we collect several soil probes, as Christine is using in the photo there, or augers is what we tend to be using more commonly. We may collect 10 or 12 of those across those 2 tractor rows, and then the composite means we put it all in a bucket and mix it up. And so we get one sample from each of those units, but it's a mixture that's been collected. So I think of it as trying to sort of capture the average of the soil in the space. And we also have been collecting core samples for bulk density on a semi-annual basis. And we collect the soil from two depths, but it's really the surface of the soil. So we have a 0 to 10 centimeter depth and a 10 to 20 centimeter depth. And I believe a lot of the projects in the Healthy Soils program are collecting from these depths, which is good because then this makes the data transferable across the program. And then, we're collecting from the surface really because that's where we expect to see the changes in the time frame of a few years on the project. So these are surface soil samples. And then I threw this photo in just to give you an idea of when are we collecting these samples. So ideally we're collecting these in the dry down period of the spring, really just before the vineyard team is ready to get the tractors in there and do the tillage. When the soil's just a little bit too moist for tillage is the ideal time for collecting soil samples. And so in this photo, you can see, there was a nice winter cover crop that got hit with the flail mower and turned into a very nice layer of mulch. And so this is just, wonderful timing where the top side of the mulch is dry and the bottom side is still got some moisture in it. And so that's what we're sort of shooting for is the late spring dry down period. I would say that's one of the ideal, maybe the ideal time in my mind to collect soil samples in California. And then what are the measurements that we've been doing on these samples? So on semi-annual basis, we've been collecting bulk density, which is a measure of the density of the soil and necessary to tell us how much carbon and nitrogen and things we have in by mass in tons. We've been measuring total carbon and total nitrogen every year, which is part of the soil health program. And then on certain years, we've been doing a full soil health analysis package with an analytical lab at Oregon State University. And so this is available. Oregon State University makes this available to producers and researchers. And so it includes all of these tests, the chemical nutrients and pH, aggregate tests, aggregate stability, active carbon, and so I don't have time to get into the details of all of those, but these are all different ways of looking at the condition of the soil and comparing it between our treatments. And then the 2020 data that we'll look at this morning just really has total carbon and nitrogen analysis. And we'll be doing next year will mark 3 years that our treatments have been in place and 3 growing seasons. And so we plan to do a larger sampling next year at the 3-year mark and run this soil health package again. And there you see our soil samples from the project drying. It's become our tradition that we're drying the samples in the greenhouse at SRJC before we send them to the lab. And so just some baseline data from the site from 2018. And I think these are all data actually from the tractor rows. And so soil organic carbon is, approximately the relationship between carbon and organic matter, which people are probably more used to looking at in a soil test, is that carbon makes up about half, a little more than half of your organic matter. So this would be a little over, this is almost 4.5% organic matter in this soil, or 2.2% carbon. So it's a soil that had a nice level of organic matter and is in very good condition to begin with, and that's a low density, which we would like to see. So it's a very nice soil, a very productive soil, and that was our starting condition. So jumping into some data from this year, so now I'll show a series of graphs that are all from the data that we got for this year. And so, one of the results, I presented something similar to this slide last year as well, is that there's a big difference in the carbon and the organic matter between the tractor rows and the vine rows at the site. And so these results are actually statistically different. I spent several days in the last week doing some statistics, and so there's a strong difference, right? They're strongly different, and it really has to do with just the cover cropping a lot of the active management of the soil and the inputs of carbon are going into the tractor rows, whereas the way that the vine rows are being managed is to keep them clean, to reduce competition with the vines, and so they're not getting the same level of inputs. And so this is one big difference in the site. And then statistically, our blocks, the two vineyard blocks, are also different. The soil is a little bit different, particularly in the northwest corner of the site. It becomes a little bit of a sandier soil, but from an experimental point of view, this is good. If you're going to use experimental blocks, you're understanding that they're different, and so it's good to confirm that. But so, one major result, big difference between tractor rows and vine rows in the project. And then tillage treatments. So then this was our first treatment. These are our tillage treatments. And so the results, I should have mentioned in the previous graph, these graphs show you both depths of soil. And so with the tillage treatments so far to date, there's no difference statistically between these tillage treatments. You can see a little bit of trend in the data toward no-till holding a bit more soil carbon. But at the moment, these things haven't separated themselves so far. And so these data are 2 full growing seasons in, right? We had the full management 2018, full management 2019, and then I got out there and pulled these samples at the beginning of the season this year. So that no difference so far in tillage treatments. And then we have the test of the compost versus the no compost. And so I'll say for me, this is the most exciting slide in the presentation, and the most exciting result for me today. And that is that there is a statistical difference after 2 years between plots that are receiving compost and plots that aren't. So already after just 2 years, the compost application, 5 tons per acre, is raising the soil carbon levels in the 0 to 10 centimeters depth, right? We have a measurable change. So this is exciting. I've taught in soil science. I was taught, and I teach others, that it takes 3 to 5 years to see the effect of a change in management. And so we're early on here, getting a good result. This also gives me confidence in on our experiment that we're going to see changes in the data. So this is exciting to me, hopefully exciting to my colleagues. And I would say it was a strong statistical difference, right? No question that this is a measurable change. And then soil nitrogen, also higher. This total soil nitrogen metric that we have here is correlated with organic matter and carbon. And so, both of these compost, it's the top 4 inches or 10 centimeters. Where it's higher, a little bit higher with nitrogen. And to back up to the carbon, this is about a 15% difference. There's about 15% more carbon in the compost treatments than the non-compost after 2 years. All right, so I'm going to move towards wrapping up. This is just a quick shot of some data from this year. I did want to share one related research article. I've been immersed in this work with the RCDs and others for the last few years. And so a very exciting study for me this year from UC Davis was this reference here. If you Google the author's name and look up deep soil inventories. And so they did sampling to, I think, to 2 meters depth in soils in the Central Valley. And basically what their paper showed was that in these annual cropping systems, that when you added compost application, that allowed for significant carbon accumulation. And just doing cover cropping didn't. And so I think, this is to say, coupled with our data, I think that personally, my personal professional opinion is that compost is really a necessary practice if we want to move the needle on soil carbon and soil organic matter in a short time frame. Compost just kind of is giving the system an extra input and an extra boost to move up. And so I think our data is pretty well in line with this. And this was a longer term study, I believe from the Russell Ranch, which is UC Davis's experimental farm. And so then I threw these pictures in here that Anya took in February. So that's a no-till plot that did not receive any compost. And then this is the plot right next to it that did, right? And so that's not so scientific, but visually, there's probably also a factor where you're getting more biomass out of your cover crops and more of an input and more of a biological response with compost. And so I'm going to wrap up there. I'll be happy to answer questions later. And yeah, I just want to express my thanks and gratitude to everyone that's made this happen, to Jackson Family Wines, to the Vineyard team for incorporating this work into their management of the site, to everyone at the RCD that makes this happen for us, to the CDFA. Cold Creek Compost has provided us with compost. Gloria manages the Soil Health Lab at Oregon State. And then everyone from the RCD and my students that have helped to collect and process soil samples, because it's a lot of soil we've been collecting. So with that, yeah, thank you. Thanks for your attention today. And I'm very encouraged by the results that I presented to you, particularly around compost.
All right, thank you, Josh. That was a really nice, great snapshot into what we're seeing so far. It's definitely exciting to look forward to next year when we're gonna redo the broader sampling to see what kind of insights we might be able to get from that. And definitely very exciting to start seeing changes already, even after 2 years of practice implementation. I did see really quickly, there was a question asking to clarify the tons per acre compost application rate. And that was, we're shooting for 5 tons per acre application rate annually. All right, and now we will transition to our partners from Jackson Family Wine. And we have Sean Kajiwara and Julian Devroe that are going to give us some insights into how this project plays a role into some broader ideas about sustainability and just their vision as an organization looking forward. And to tell you a little bit more about both of them, Sean was born and raised on the Hawaiian island of Oahu, where he gained invaluable farming experience from the family fruit farm and nursery. His first job out of college was actually a harvest lab internship with Jackson Family Wines. After the internship, Sean transitioned out of the lab and into the agricultural fields, and today he serves as Jackson Family Wines Director of Farming, overseeing operations across the company's Russian River Valley, Sonoma Coast, and Anderson Valley vineyards. Sean has a degree in biochemistry from the University of Washington. And Julian Devroe is the Director of Sustainability at Jackson Family Wines. His focus is on setting strategies and implementation of water and energy efficiency, as well as waste diversion projects throughout the company. He also works closely in communicating Jackson Family Wines' commitment to sustainability to their customers and broader corporate partners, and obviously also collaborates with partners like the RCD on research and explorations of other opportunities within their vineyards. And Julian holds an MBA in Sustainable Management from the Presidio Graduate School in San Francisco. All right, and I will pass it off to you gentlemen to share some insights from the organization.
Thank you, Anya. This is Julian. I'm not on video right now. I will be shortly, but Sean is going to kick us off with some of the observations that he's seeing in the vineyards at Saralee's through the trial. Can everybody see my screen?
Yes.
Excellent. Take it away, sir.
Thanks, Anya. Thanks, Julian. Julian, can you turn to the first slide, please?
Hang on. There we go.
I wanted to give everyone a little more background on the Saralee vineyard. So the Pinot Noir is clone 777 and the Chardonnay is clone 96. Both are on 5C rootstock, so fairly vigorous rootstock. Planted in 1998, so we're going on just over 20 years. So these vines are quadrilateral cordon. Spacing is 12 feet by 6 feet. The soil is a sandy loam, so the roots are going down about 5 feet into the soil. And once you get down about 5 feet, you reach pretty much what is pure sand and water. So these vines are very happy, they're vigorous. That's one of the reasons why I chose this vineyard for the trial, because if we're not going to till, I wanted to make sure that I'm still producing a healthy crop and that the vines are still healthy. I didn't want to do this project at a place like Bellflower, which is just down the street from Saralee, but at Bellflower Vineyard, you have this hard pan about 2 feet down from the topsoil, and we rely heavily on tillage there. And probably the rootstock there is also the wrong rootstock chosen for that property. So a weak rootstock mixed with weak soils, it's really difficult to have that competition with the cover crop. So that's one of the main reasons why we picked Saralee, was that these vines are fairly healthy, and we wanted to really look at keeping this cover crop and seeing if that would benefit us with soil health. A few years back, I started eliminating herbicide use from the vineyards that I farm. And I think one of the main reasons why I did that is because, maybe 5 years ago, I started looking at soil analysis, and I started seeing just these huge numbers of nematode populations in the soil, and they were all, really bad nematodes like dagger and root knot. And I was wondering, like, why do we have, these really high populations of these pests? And I was taking a look at our farming practices of, just spraying the berm every year with chemicals, and just a lack of biodiversity in the vineyard. We already have a monoculture of grapes that we're planting. My thought was that if we could increase biodiversity in our cover crops, that perhaps we could, lower nematode populations, maybe create a more diverse ecosystem in the soil to combat, those high nematode populations. And so, I think when you saw Josh's slides, you could see the clean burn underneath the vines, and that's because we used to use herbicide at Saralee. Now we don't use herbicide. It takes some time to get this right. You have to get implements such as weed machines. We use a Clemens here at Saralee. It's a blade that pretty much just cuts the weeds just below the soil surface. This picture here was taken this week, and so we had gone through this vineyard twice this year with the Clemens, but you can see the weeds coming back under the vines in both photos. But we'll have another pass here soon enough, and it'll be clean again. I'm still looking at other implements such as the sunflower, which has a mowing head attachment. So we're looking on a vigorous vineyard like this. Perhaps we're not going to have to till at all, or even do under row cultivation. Maybe it's just going to be just all mowed. And so my hope is that having this cover crop here is going to help us bring that biodiversity to the soil. So these photos compared, alternate row tillage compared with no-till plus compost added. It's a little hard to tell what's going on here because we have gone to this this no herbicide regime, and so pretty much this is the new look of our vineyard now. I think we're happy about it. Julian, could you go to the next slide, please? You know, before, in the past, I used to really want that clean burn, and also, you know, our practice in the past used to be let's till every other row. But now, you know, I can't see much of a difference between the vigor in the canopy, between no-till, alternate tillage. So, and I also don't see any like yield difference, major yield difference or quality difference. We're averaging about 5 to 6 tons an acre on this vineyard. So I think going forward at a vigorous site like this, we'll probably continue to you know, do no-till, and also you know, start mowing the cover crop underneath the vine rather than tilling it. I think compost is a really important piece to this. We used to drip you know, chemical fertilizer through the drip system, but we also stopped doing that too. Now we just broadcast the compost throughout the vineyard. And you know, there's some managers who like to just put the compost right under the drip emitter, or you know, just ban the compost along the vine. But to me, you know, we're trying to build the health of not just the vines, but of also the cover crop as well. That's contributing to the this biodiversity in the vineyard. And hopefully what we're doing here is just creating a more resilient vine, and also a soil that's going to, you know, help these vines live a much longer time. So, Julia, next slide, please. Yeah, so here you can see that you know, the canopy this is a photo taken this week. Canopy is looking really good. You know, this is going to probably be a lighter year here for us in the Russian River, but still around 5 ton an acre. So things are looking good. Julia, next slide.
I think that's it for the slides. That's the alternate year row right there on the left now.
Okay, great. And one other point on cover crop is, in the past, I used to use, you know, just a simple mix of barley, rye, and crimson clover. And, you know, it was affordable. It did the job with erosion control. But these days I'm putting in, you know, like 14 to 16 different seed types, you know, different annuals, brassicas, beans into the cover crop when we seed in August. And that's to just build more biodiversity in the vineyard. And so the hope is that, you know, with this biodiversity, we're just going to create this better soil health. And if anyone has questions, I'll take them after Julian presents.
Great. Thank you, Sean. So I'm going to zoom out a little bit from what Sean just presented at Saralee's and the work that we're doing with the RCD and CDFA with the Healthy Soils Project into where we're ultimately going in the future. And, you know, I think as Sean describes the activities that are taking place in the vineyards that he farms, and it's not just Saralee's vineyard, but it's you know, he's kind of on the vanguard and on the leading edge for Jackson Family Wines as we make this transition. So we're seeing a lot of this happening, you know, in the Russian River, in Anderson Valley, and on the Sonoma coast. We are really interested in that intersection between soil health and high quality wine growing. and also kind of the mutual benefits where we can potentially increase soil carbon sequestration within our planted vines. We're also looking at it in our unplanted lands. So for every acre of vineyard that we have at Jackson Family Wines, we have over 1 acre of natural and wild lands that we're really kind of seeing as a vital part of not just healthy grape growing, but also opportunities to kind of continue to preserve and promote biodiversity and enhance soil carbon sequestration. We recently went through about a 6-month internal goal setting process over the first half of this year, which was abbreviated a little bit with COVID and everything that happened with the shelter-in-place orders. But we were able to do a lot of amazing work and a lot of internal kind of alignment on where we want to be as a company 10 to 30 years down the road as it relates to some key resource applications. And we've identified some high-level goals that we're going to be starting to talk about a little bit more publicly moving forward. And, you know, from a very high level, we've committed to becoming climate positive by 2050 from a greenhouse gas emissions standpoint across our entire emissions value chain. So scopes 1, 2, and 3, all of our direct and indirect. And that's an important point, because as a land-based company, we are recognizing through the research of people like Dr. Beniston and the Sonoma RCD and the Healthy Soils Program, that soil carbon sequestration can have a tremendous opportunity to help us achieve those goals as an ag-based company and as a farming company. And that so our ability to trial and test these methodologies in a statistically significant setting is crucial to our future management activities. So as we grow and as we scale, we are going to be working with you know, the activities that Sean is doing and looking to kind of broaden those and expand these trials across all of the regions within which Jackson Family Wines farms, from Santa Barbara County in the south all the way up to the Willamette Valley in Oregon. So we've got an amazing opportunity here to really understand the different parameters for soil health and how that relates to soil carbon sequestration, quality, and all of the other touch points that we are really going after here. So there's an amazing opportunity, and as we look bigger picture. Soil carbon sequestration is important, obviously, from a reversing and a mitigating climate change standpoint, and also enhancing just resiliency in the face of climate change. What we haven't really talked too much about, and I think Dr. Lozcano is going to be probably touching on some of it, but it's also, you know, appropriate soil management really enhances a vineyard's ability to hold water and retain water. So we're seeing kind of all these mutual benefits that we're excited to scale up in our trials over the next few years to really fully understand if we can scale this to a full degree across all the vineyard acres that we farm. And from there, you know, beyond just the benefits of soil carbon sequestration, we see the potential development of market forces that will help pay farmers to sequester carbon in the soil. And as more and more companies and more local and regional governments start to become serious about climate action and the importance of achieving climate neutrality and carbon neutrality over the next 10 to 25 years, we see tremendous market opportunities that are going to emerge that will actually help pay farmers to capture and store carbon in the soil. And a lot of this work is being pioneered, like the Sonoma RCD, as well as some of the agencies here in California with CDFA and this whole program. So as we start to get to understand the opportunities, we are really excited to try to figure out ways in which we can modify and adjust our farming practices to be able to participate and to lead this change. So it's very, very exciting stuff. And then finally, I just wanted to put in a shameless plug for those of you who are interested in climate action and leveraging the power of the wine industry to really take a leadership role in addressing the climate crisis. We recently, in collaboration with Familia Torres, which is Spain's preeminent family-owned wine company, we recently launched International Wineries for Climate Action, which is a group that's open to any wine company who believes that the climate crisis is something that deserves urgent attention. And what we've done is we've kind of created this this consortium of member wineries that are taking an aggressive approach and a science-based approach to measuring our greenhouse gas emissions footprints across all of our value chains. So that's scopes 1, 2, and 3, and then setting those targets for achieving climate neutrality by 2050, with an intermediate goal of a 50% reduction in the next 10 years. And we see this group as a great opportunity for kind of best practices sharing across different wine growing regions, different continents, different countries. So today we are 8 members strong. We have wine companies now representing 4 different continents. And this group is open to anybody who's interested. So if anybody is sitting at home wondering what they can do and what the wine industry can do to really take decisive action, we would love to connect with you and share more about International Wineries for Climate Action. And in closing, I just, I want to recognize everybody who's worked with us on this project. There's a tremendous amount of research that has gone into this and a tremendous amount of work, and we're excited to continue to learn and to start to apply some of these learnings at a larger scale across more of our vineyards. So I want to tip my hat again to Sean for being on the leading edge of this here at Jackson Family Wines and really continuing to promote these activities internally as we try to figure out how we can adopt and scale. And all of the partners that we've worked with, Dr. Beniston, Sonoma RCD, CDFA, Cold Creek Compost, who's provided compost for this trial free of charge. Everybody who's really helped kind of further the learnings on this. It's been a tremendous partnership, and we're excited to see what happens over the next couple of years as we get additional soil data back. So with that, I will turn it over.
All right, thank you, Sean and Julian, for giving us some really good insights about some of the bigger and longer term thought processes that are happening at Jackson Family Wines, and how you're using the information we're gathering out of this demonstration trial to sort of set some really cool goals for yourselves. So I will again remind everyone to please add your questions into the Q&A window. Just for the sake of being able to move through the presentations and have an opportunity for everyone to hear our great next presenter. We're going to hold the questions until the end, but Sean and Julian will be available, as well as Josh, to tackle the questions that you've already entered for them. So stay tuned for the Q&A. And in the meantime, I would like to introduce Dr. Cristina Lozcano. She is an assistant professor at University of California, Davis. And Dr. Lozcano's research focuses on how soil organisms interact with each other and their abiotic environment to regulate ecosystem processes such as nutrient cycling. Dr. Lozcano started working in vineyard soils about 4 years ago, investigating how soil management can affect soil biota, carbon and nitrogen cycling, and grape quality, which is something she will be touching on today in her presentation. She completed her PhD at the Soil Ecology Research Group from the University of Vigo, Spain. Hopefully I'm pronouncing that right. Afterwards, she worked as a postdoctoral scholar at the Technical University of Denmark, the University of Calgary, and UC Davis. Before joining the Davis faculty, though, which happened in 2019, Dr. Lozcano was an assistant professor of soil ecology at the California Polytechnic State University in San Luis Obispo. So we welcome Dr. Lozcano as our keynote speaker here to share some really interesting insights from her own research and research from other studies that talk about wine grape production and how healthy soils can play a role in that. So take it away, Dr. Lozcano.
Thanks, Anya. So first of all, can everybody see my screen? Can everybody hear me okay?
Yes.
Okay, perfect. So I hope there are no technical difficulties. You know, sometimes my neighbor just decides to walk at random times in the morning. So hopefully it doesn't happen today and everything goes smoothly. So again, thanks for inviting me. It's really exciting to be here and to listen to all the research going on. And this is one of the things that actually attracted me to work in vineyards. I just started working with vineyard soil health 4 years ago, but it's really an exciting field, and I'm really impressed about how the industry is really ahead of the game and active in incorporating all of these sustainable soil management strategies. So today in my presentation, I want to talk about some general concepts of soil health, the definition of soil health, and some basic aspects of that definition. But I want to focus as well on how much we still need to learn, specifically on the meaning of soil health for wine grape production. So what are the knowledge gaps? What do we need to work on? And I'm also going to show some of the projects that I've been working on for the last years, and some new projects also that we just started. So before that, I'm hoping that this is going to be a relatively, oops, sorry, okay, there we go. Now it's a little bit slow. Okay, so I hope this is gonna be relatively interactive. So I prepared a poll. This is an online polling tool where you can either respond through the website or through text. So you can pick up your phone and text soil ecology altogether, soil ecology 428 to this number, 22333. So after you do that, then you are in the system and then you can text your answer. So in this case, my question to you is, who do you identify best with? Are you a vineyard manager? Are you an owner of a vineyard? Are you a consultant, a researcher, a student, or other? So I'm gonna give you a minute and see if you can send me your answer. So we have a little bit of an idea of who is there listening. And again, oh, cool, we got one, it's working, great. So you can also, again, log into the website, paulf.com/soilecology428, and also send your answer through there. Or use your phone and text to this number, text soil ecology 428 first, and then your answer. Okay, so we got vineyard managers, researchers, others, okay. And now I'm curious about the others. I'm assuming there might be people working for RCD or maybe an RCS. Great, consultants. We don't have any vineyard owners. No students out there either? Okay, there we go. We got a vineyard owner. There we go. All right, well, cool. Let's move on then. Now I know a little bit more about you. Okay, so to the topic. Over the recent years, there's an increasing awareness by the general public, by the researchers, government organizations, non-government organizations, on the role of soils as essential and non-renewable resources that sustain not just crop production, but also environmental quality. And they actually have a very important role in climate change mitigation by sequestering carbon and regulating the emissions of greenhouse gases. So I really like this picture here because it's showing the tremendous amount of diversity in soils, so diversity below ground, but also diversity above ground. in land uses, landscapes, and also the diversity in the people that depend on soils. So because of this increased awareness, there are also several international initiatives, national initiatives, but also local and state initiatives that are trying to promote soil conservation and the preservation of soil health. And we all know, we heard about today about the CDFA Healthy Soils Program. This is a program that was started in 2017. And the goal of this program is to support farmers and ranchers to adopt sustainable soil management practices that at the same time enhance soil health by increasing soil organic matter and promoting carbon sequestration. But also on the other hand, they reduce the environmental impacts associated to crop production by reducing greenhouse gas emissions. All right, so this program provides subsidies to growers, to farmers and ranchers to incorporate these practices at no cost. So what do we mean by soil health? And this is a definition that you have seen already probably several times, but there's a reason why I'm revisiting this. And it is because it's a very general definition that is actually kind of vague when it comes to applying it to specific crops, like wine grapes. So the definition of soil health by NRCS says that a healthy soil, soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So basically, a healthy soil is a soil that functions and that provides services, right? But those services need to be tailored to the specific conditions and crops, right? So there are several open questions in regards to soil health and wine grape production, and this is what I'm going to focus on for this presentation. So first of all, we need to define what soil health means for wine grape production. What are the most important properties of soils that are leading to the production of high quality grapes? What are those aspects of soil that we need to target through management, right? And we also need to determine the importance of new soil aspects, like for example, soil biota, soil carbon, and the soil microbiome for wine grape production. And we need to revisit the concept of terroir. And finally, once we have all that information, management practices need to be tailored to those specific conditions and requirements of wine grapes. So going back to the definition of soil health is relatively new, and it's a concept that it has been embraced both by the industry, by the general public, by researchers, but it really is not that new. It builds on a more like an older concept, which is soil quality. And the concept of soil quality, it kind of actually refers to properties that soil have, such as, for example, a certain amount of nutrients, certain soil fertility, or a certain soil texture that are good for a certain purpose. On the other hand, soil health and the concept of soil health emphasizes not only the properties of the soil, but the functions that lead to those properties, that support those properties. So, and here I wanna remark two important differences with soil quality. First of all, this is a dynamic concept. It involves processes, and therefore it's a concept that needs to be maintained. Those are functions that need to be maintained, and that they are very directly affected by management, right? They're sensitive to management. And the second thing that I want to highlight is that a lot of these functions and processes are actually supported by soil biota. So then in this new framework, soil is actually considered as an ecosystem. And I really like this newer definition of soil health by Professor Ariana Van Bruggen, which says that a healthy soil ecosystem is a system with integrity of nutrient cycles and energy flows, stability, and resilience to disturbance or stress. So now we got a little bit more precise definition of a soil health, right? We're talking about nutrient cycles, flows of energy, stability, and resilience to stress. So those functions need to be able to support those processes. So what specifically are the processes that are supported by a healthy soil? Here we got a summary of the different properties of healthy soils, and they are classified by color according to different categories. So we have like fertility related properties. Oops, sorry, this is very sensitive. We got properties related to soil physical properties. I'm going to use properties related to soil physical aspects, yields, biological aspects, and environmental aspects. So for example, a healthy soil needs to be able to supply nutrients for the crop, but also retain nutrients, right? We don't want to lose nutrients to the groundwater or to the atmosphere. So nutrients need to be efficiently retained within the soil. In terms of physical properties, a healthy soil will store water, but at the same time, it will not get flooded, so it will have good water infiltration and drainage. A healthy soil will support good yields, but also the quality of the crop. It will hold diverse soil organisms, but also those organisms will be active, it will be functioning all the time and supporting all of the processes. And in terms of environmental properties, a healthy soil will reduce emissions of greenhouse gases and it will store carbon. How do we measure these functions? Well, we got what it's called soil health indicators. And those are actually measurable outcomes that will be indicative of the amount of the function in the soil. So for example, for nutrient retention, we can measure the mineralization rates, nitrification rates. For nutrient supply, we'll measure the amount of plant available nutrients in the soil at a certain time. For water storage, we measure water holding capacity, infiltration rates, obviously for yields, the amount of harvested crop per area. And in terms of crop quality, this is more specific to crop, to depending the type of crop, right? So for grapes, maybe we want to look at specific phenolics or breaks or acidity. Other crops, we look at vitamins and micronutrients. For diversity, there's different genetic markers that will tell us the amount of the different types of organisms that live in the soil. For activity, we usually use respiration, microbial respiration, and the release of CO2, but we can also use enzymes. And for greenhouse gas emissions, we measure the flux of nitrous oxide, CO2, and methane. And for carbon storage, we measure directly the amount of carbon that the soil has. So one important thing that I want to highlight here too, is that obviously the properties and indicators of soil health that are targeted through management need to be weighed differently depending on the crop, but also depending on the production goals and site-specific challenges. All right, so all of these different indicators of properties are not emphasized the same for different conditions. And I'm going to explain why. For example, think about corn and wine grapes. They're extremely different, right? So the properties of soils that you want to emphasize and that you want to look for are also different. And that means that the meaning of soil health, it's different for corn, for growing corn, than for growing grapes, right? So now I got another question for you. Okay, pick up your phones And so my question is, given your experience and what we just learned about soil properties and soil health indicators, what do you think is the most important soil property for growing wine grapes? And just text any words again to the same number, 22333, or through the website paulebb.com/soilecology428. Send any word. This is going to generate a word cloud. So the words that are texted the most will appear bigger, and the words that are texted the least will appear smaller. Okay, great. Here we go. Infiltration, balance, micronutrients, microbes. I like that. All right, so far we can a very even distribution. pH. Somebody mentioned pH. Microbes. Again. Balance. That's a big one then. All right, more. We need more. Anybody else? All right, so we got a good idea here. And I like the word balance. I really like it. We have a bit of a mix between physical properties like infiltration, permeability, the depth of the soil. We got some biological, mycorrhizal, microorganisms, life, fungi. Good. I like that. So it's kind of a mix between micronutrients. So we got some chemicals. All right. And the word balance is very big. I really like that. Okay. So moving on, going back to that comparison of between corn and wine grapes. So those are very different crops. So the priorities and the soil properties that we need to manage and emphasize are also going to be different, right? And not just because crops have different requirements, but also these two crops are also grown in different areas, which have also different challenges, right? So corn is grown primarily in the Midwest. Corn is a crop that has very high nutrient requirements. And what growers are looking for really, it's just high yields, right? However, in wine grapes, we want not just yields, but growers are also interested in a specific chemical profile in the grapes. So quality is also emphasized, right? So we don't want so much a crop that is producing high yields, but a crop that is producing a certain chemical profile. In corn, in one hand, to produce these high yields, what we need, it's a lot of nutrients, right? So we need to have a soil that provides the nutrients that corn requires, and especially a lot of nitrogen. Corn has very high nitrogen requirements. But also, we don't want to lose that nitrogen. We don't want to have high leaching rates or emissions of greenhouse gases. So we want a soil that holds the nutrients, retains the nutrients. How about wine grapes? Well, we're not so interested in having a high supply of nitrogen, right? Because it can actually be counterproductive for wine grapes. But we are interested, on the other hand, on having a soil that retains the nutrients, that doesn't lose nutrients and recycles them. And also being California, all right, and the problems we're having with drought, we want to have a soil that stores water. So the definition of a healthy soil for growing wine grapes in California looks very different than the definition of a healthy soil for growing corn in the Midwest, right? And it turns out that most of the soil health indicators and definition of soil health have been defined for crops in the Midwest, but not for wine grapes. So this is something that we are working on right now, and finding a better definition of soil health for wine grape production. And this is a project that the graduate student, Noelimar Gonzalez, is doing right now. She's starting to carry out semi-structured interviews with growers and vineyard managers. And she's also going to do soil sampling through Napa Valley. So we get information on what are the most important things that growers look for in soils to support soil health, but also high quality grapes. This is a really exciting project that we just started a month ago, and it's supported by the American Vineyard Foundation, and it's done in collaboration with Napa Valley Grape Grower Association. We are looking for collaborators, so if you want to participate, please send me an email to clozcano@ucdavis.edu, and we'll follow up with more information on how you can help us with this project. All right, so the importance of soils for wine grape production is nothing new. This is something that is kind of incorporated into the concept of terroir. The concept of terroir defines the importance of soil together with landscape, climate, topography, and the grape variety for wine production, right? And for wine quality. So this is nothing new, and there has been a lot of research going on for several years, right? However, the aspects of soil that are emphasized in the concept of terroir are slightly different from the aspects of soil that are emphasized by this new concept of soil health. And so here, this picture kind of shows a little bit how the two concepts intersect and overlap with each other. So things that are incorporated or included within the definition of terroir are, for example, physical aspects of soil, like the depth of the soil, so things that you mentioned before, structure, texture, but also the mineralogy, cation exchange capacity, pH, nitrogen, water holding capacity, etc. Now, the concept of soil health, it's similar, but it's not exactly the same. Some of the properties of a healthy soil are also incorporated into the concept of terroir, but there's new properties that are considered by soil health that haven't been considered for the concept of terroir before. Those properties are specifically the soil biota, microbial biomass and activity, biodiversity, and also soil carbon. So now my question is, given the important role of soil carbon, microbial activity, and soil biota in general for soil health, do we need to redefine the concept of terroir to include these soil properties? And then the second question is, how is management of soil health improving the expression of the terroir, right? So those are things we need to work on. And those are topics that my research group is working on at the moment. So starting by the soil microbiome. How is the soil microbiome important for wine grape production? Well, the soil microbiome supports really important functions in soils. For example, the cycling of nutrients, the control of diseases, the balance between different species in the soil is extremely important for keeping diseases in check. Soil microorganisms are responsible for decomposition, so they are very important for carbon sequestration in soils. And we have to remember that wine production, it's primarily a microbial process. So soil microbes are very important as well for wine production and for fermentation. There's a lot of new and very interesting research coming out that it's showing that there is a connection between the soil microbiome and the properties of the wine, the microbiome in the grapes and the properties, the chemical profiles of the grapes and the wine. So how is that happening? Well, microorganisms in the soil can actually be transferred to the grapes either through the vine as endophytes, so they can move inside the vine and be transported all the way to the leaves and to the grapes. But also microbes can be transported in dust particles, in raindrops, or even in the hands of the people picking grapes, right? So there have been studies recently showing that there is a connection between the types of microbes you find in the grapes and the types of microbes in the soil. And that is also very strongly associated to specific geographical areas. So people is talking now about the concept of microbial terroir. Now, this is something, it's a very exciting new field of research. It's very promising. But there's also several things that we don't understand in regards of to the microbiome and wine grape production. We need a better understanding on how management of the soil it's affecting the soil microbiome. And also we need a better understanding on how this whole soil food web, not just the microbes, are affecting the quality of the grapes and the quality of the wines being produced. And why the whole soil food web? Well, it turns out that the whole diversity of organisms that live in the soil actually are very important drivers of the microbiome. The food web, it's a concept that describes all of the organisms that live in the soil and how they interact with each other. And by interacting with each other, they regulate the ecological processes that are happening in the soil, such as, for example, decomposition of organic matter, nutrient cycling, or even the formation of soil structure. So a soil food web, as you can see here, this is a simplified food web, but it kind of represents the basic levels, which are here defined as trophic levels, right? So in a food web, a trophic level constitutes the feed for the next level, So in this case, we have the first trophic level, which are the primary producers, usually plants. Plants fix carbon from the atmosphere with the energy from the sun. They generate organic matter, which is integrated into the soil. And then this is a food source for the next trophic level, the second trophic level that has the decomposers, the microbes like bacteria and fungi, but also has plant parasites, herbicides, like for example, the nematodes here. Well, these are food sources for the third trophic level, which includes shredders, predators, and grazers. And their third trophic level at the same time is a food source for the fourth trophic level, which has predators, and the fifth trophic level has higher level predators, all right? So by feeding on each other, they modify the activity and the functioning of all the different organisms, right? So for example, microbial grazers, we have here in this level, by feeding on bacteria and fungi, they're extremely important in regulating the structure and the function of soil microbes. So really, to consider soil biota, we should look at the whole food web, not just the soil microbes. But looking at the food web, it's a really daunting task. It requires a lot of knowledge and very specific knowledge to be able to identify every single organism that lives in the soil. It's almost impossible. So what we do is we use proxies. So there is one type of organism in this food web that is represented in almost all the different levels. Maybe you have already identified, but if you look carefully, nematodes are actually represented in almost every level of the food web. We got root feeders, which are herbivores, and they feed directly in plants. But we also have bacterivores and fungivore nematodes. We got predator nematodes that feed on these ones, on the fungivores and bacterivores, all right? So just by looking at nematodes, we can have a really good idea of the structure and complexity of the food web. And this is what we've done in a field study that we did a couple of years ago in a vineyard in Edna Valley in San Luis Obispo. So we wanted to look at the distribution of nematodes and the complexity of the food web across different places in the vineyard. So we took soil samples from the tractor row and from under the vine. And we saw something really striking. So the tractor rows have way more nematodes than the vine rows. The abundance of nematodes is much, much higher, as you can see here. Not only that, but there were differences on the food web structure. So the type of nematodes that we had in the two different locations were also different. And here you have this pie chart is showing the proportion of the different types of nematodes in the tractor row here and the vine row down here. So in the tractor row, we had a better proportion of the different types of nematodes, so a more structured food web with more fungivores and bacterivores as compared to herbivores than the vine row. The vine row had less nematodes, but in the vine row, most of the nematodes were actually the bad guys, the herbivores, all right? And in addition to that, there were certain soil health indicators like total soil carbon, active carbon, or pH that were also very directly related to this abundance and, of nematodes and food web complexity. So carbon, active carbon was higher in the tractor row together with this more balanced soil nematode food web. So what we're doing at the soil, the management of the soil in the tractor rows, having the cover crop and the carbon input, it's having a direct impact on soil biota and the soil food web, therefore affecting potentially the microbiome and soil health in general. And that leads me to the third topic I want to cover in this presentation, which is the management strategies that we choose for our vineyards and the effect that they have on soil health. So as you probably already know, the principles of management for healthy soils are three. First of all, increase soil organic matter. Second, reduce disturbance of the soil. And third, increase diversity by maintaining a living plant cover in the soil. So usually the practices that most people is using, it's compost application to, it's a direct input of soil organic matter to the soil. To reduce disturbance, people is transitioning to a no-till or reduced till. And to increase diversity and keep a living plant cover, many vineyards are incorporating cover crops into their management strategies. So I got another question for you, and this is the last one. So if you're a vineyard manager or you're working in a vineyard, are you currently using any of these practices, or are you using a combination of these practices? Are you using compost? Are you using no-till, cover crop, or a combination of compost and no-till? Like we've seen in the Saralee vineyard. Are you using compost and cover crops? Are you using no-till and cover crops, or the three, compost, no-till, and cover crops? Wow, we got a lot of answers on the combination of the three. That's really good. Good variety. Still some answers coming in. Okay, more for compost. All right, so I really like that people is actually using a combination of practices, compost combined with no-till and cover crop. Very good. All right. So there is a lot of research that has been carried out over the last 20 to 30 years that show that these practices are really effective at increasing soil organic matter and soil carbon. And so there is a very good knowledge, and we know that these practices contribute to, by increasing soil organic carbon, these practices contribute to mitigating climate change. We know that soil organic matter has approximately 60% of carbon. So putting organic matter into the soil by using cover crops or compost, it's putting carbon into the soil directly. And so just to give you an idea of how much greenhouse gas emissions we can mitigate by using these practices. In California, soils have usually very low amounts of soil organic matter and low amounts of soil carbon, but a single 0.1% increase in carbon per acre, it's equivalent to removing the emissions of an average car for a whole year, right? So increasing soil carbon by 0.1% per acre mitigates the emissions of an average car in 1 year. So we think about all the 45,000 acres of wine grapes, for example, in Napa. And so increasing soil carbon by 0.1%, if everybody would incorporate healthy soil practices and would achieve a 0.1% increase, then that would remove the same amount of cars, right, from the streets. It will be equivalent to removing the same amount of cars from the streets. Now I saw that Josh in his presentation showed a 0.3 increase in carbon sequestration with compost application, which is really good. So you can imagine the benefits of using compost. A second benefit that is also pretty well known, it's the contribution to the adaptation to climate change. And especially in California, we know we have went through like pretty severe droughts. We know that water scarcity is going to be a problem in the future. So improving soil physical properties and the capacity of soil to hold on to water is extremely important. So increasing soil organic matter improves the capacity of the soil to hold on to water. And there are some studies out there that show that for every 1% increase in soil organic matter, that can result in up to 20,000 gallons of available soil water being retained per acre. That's a lot. And this number actually received a lot of criticism. And the reality is that, yes, this is a best case scenario, but the reality is that there are a lot of uncertainties. And in order to be successful at applying these healthy soil practices, we also need to create...
Yeah, maybe we just...
Sorry? Somebody, okay. So in order to effectively integrate these practices into agriculture, we need to create realistic expectations on the outcomes. And the reality is that for these practices, compost application, no-till all cover crops, we don't have a single soil health outcome, but we might have different scenarios depending on the context or where we are applying these practices. So the increase in soil organic matter after applying compost can be different if we are on a sandy soil or if we are on a clay soil. And that also can have different effects to grape quality. All right. For example, this is a study that was done by Carrie Steenworth and Kyla Burns in Napa Valley a few years ago. So they went out and sampled topsoil from different vineyards. that were using different types of cover crops and cover crop mixes. And they saw that actually the microbiome was significantly different between all of these different types of cover crops. So here are the different colors. So here in yellow, for example, we got no cover crop, grasses, legumes and cereals, legumes, cereals and mustard, legumes and mustards, okay? And the different dots here represent the diversity of the soil microbial community. The closer together, the more similar they are, and the farther apart, the more different they are. So what you see here is that grasses were pretty different from mixes of cover crops, like legumes, cereals, and mustards. So there is a different practices generate different outcomes in soil health, all right? The choice of cover crop will also have different outcomes in the microbial community and also carbon sequestration, etc. There are also uncertainties associated to the fact that many practices are usually combined. They are overlapped with each other. And a very clear example of this is how cover crops are managed. And one of the most critical management strategies when it comes to managing cover crops is the termination strategy, which can really have very strong impacts on the soil physical, chemical, and biological properties of soil. So what are the options here? Cover crops can be terminated either by spraying herbicide. This is something pretty common. Some people use tillage, mowing and tillage. And recently, several vineyards are incorporating sheep grazing as a kind of more environmentally friendly way of terminating a cover crop. All of these options have plus and cons. So a herbicide application is obviously effective, but it has negative aspects, which are the non-targeted effects on soil biota and human health. Tillage is also effective and it promotes a quick mixing of organic matter into the soil. Some people use it because they think that it promotes infiltration, especially in heavy clay soils. But on the other hand, it destroys soil structure. It may lead to issues of compaction and loss of carbon from the soil. It's also labor and fuel intensive. Grazing, it's also pretty effective. Sheep, they do a really good job at getting rid of the cover crop. It adds nitrogen to the soil through the feces. It can stimulate cover crop if it's done right, and it can stimulate soil biota. On the other hand, if grazing is not done right, it can lead to issues of compaction. There are also limitations because it can only be used during the dormant season. And in some cases, it might be a source of greenhouse gas emissions because the manure might generate emissions of nitrous oxide and methane, right? So there's pros and cons. So to study this, we're carrying out right now a field trial. This is also a study that was supported by the CDFA Healthy Soils Program. We started in 2018, so we're still collecting data. This field trial was set up at Tablas Creek in Paso Robles. And we are using a combination of tillage treatments. So you see here, and grazing treatment. So it's a full factorial design where we have no-till rows here in blue, till rows in yellow, alternating in the vineyard. And then overlapping the till and no-till rows, we got grazing plots. So we are fencing these areas and we are letting the sheep graze on the cover crop for a whole day. Afterwards, after every management event, we collect soil samples, gas samples, and also every harvest, we collect grapes to determine yields and the quality of the grapes. In the soils, we are looking at soil health indicators like total and active carbon, microbial biomass, total and available nitrogen, and physical properties of the soil like bulk density and infiltration. And we are collecting greenhouse gas emissions, methane, CO2, and nitrous oxide. This is a project that it's going on, so I don't have a lot of results to show you yet, but I do have some preliminary results in terms of total soil carbon that I'm showing you here. So this is total carbon in percentage measured at 2 depths. The surface of the soil from 0 to 6 inches, and the subsurface from 6 to 12. In orange, we got tilled treatments or tilled soils. In green, we have no tilled, and then darker colors indicate grazed treatments. So what you've seen is that pretty much there are no effects of the treatments so far in soil carbon. We do have a remarkable effect or difference rather than effect between soil carbon in the topsoil and soil carbon in the subsurface. This is something very common, but so far, after 2 years of tillage and grazing treatments, we have seen no effects on total soil carbon. And there's reasons for this. One is that this is a very heavy clay soil, so maybe the effects on soil organic matter might be not so easy to see. And maybe we need to apply the treatments for longer. So this is too short of a trial to see any effects. Something similar is happening in a second trial that we are running currently within the same project at J-Lower Vineyards in Paso Robles. In this case, we are looking at compost application and the effects of different rates of compost application. We have 0, this is a control. We're applying no compost at all. And then we have 2 tons, 4, and 6 tons of compost being applied. And so here we got the total carbon at the different soil depths. And so as you can see, there's a slight trend for higher carbon with compost application, but no significant differences so far. So again, this is very different from what Josh has seen Saralee's vineyard. And the reasons for this can be many. One of the most important reasons that drives all this variability in outcomes of healthy soil practices is the type of soil. And this is the last point I want to make in my presentation, is the type of soil where healthy soil management are applied is extremely important because not all the soils have the same capacity to sequester carbon. Clay soils are usually better and will have higher amounts of carbon than sandy soils. So what you can expect after applying healthy soil practices, it's different. And it's important to recognize that. So this is another study from Kerry Steenworth and Kyla Burns in Napa Valley, and it's showing that for all the different parts of the AVA, different AVAs in Napa, there's absolutely different amounts of carbon in those soils. Okay, so the effects of this carbon on soil health, the outcomes for soil quality that you can expect are also different, right? So in order to clarify these, we also have started within this new project supported by the American Vineyard Foundation and the Napa Valley Grape Grower Association. We're going out to all of these different soils in Napa Valley, collecting soils and measuring a series of soil health indicators to see what is the variability that we can expect in different soil types. The purpose of this is to generate a baseline of soil health indicators and variability so growers have realistic expectations on how much they can increase soil carbon for a specific type of soil, how much microbial biomass or how much nitrogen they can also expect for a specific type of soil. And so again, this is something we just started, and we're gonna be out collecting soils coming up next winter. So please, if you're interested in participating, send me an email. We're looking for collaborators. So wrapping up, we need to understand the meaning of soil health for wine grape production. And we need to set up realistic goals, and that's going to determine how effective the practices are going to be. And at the end of the day, there's no silver bullet, and it's a combination of the soil, climate, and overlapping management practices that is going to affect the specific outcome of the practices. So you need to know your soil and tailor your practices to your soil, your specific environmental conditions, the grapes that you're growing, and also the specific outcomes that you're looking for. All right, so I think I am a little bit over time. So to wrap up, we know the importance of soil health for wine grape production. We know that biota is extremely important for supporting soil health, and that supporting soil health by using healthy soil management practices increases soil organic matter, and that contributes to climate change mitigation and adaptation. But we still need to work on understanding the importance of the microbiome and soil biota, the whole food web for wine grape production. We need to understand how much can we build soil health for different soil types. We need to build realistic guidelines. And also we need to understand how long does it take to build soil health for different soil types. Okay, so that's all from me. Thank you very much for listening. And if you have any questions, I think we have a Q&A session coming up now. So I'm happy to answer any questions that you have. And if you also want to send me an email to collaborate or work with us, don't hesitate in reaching out. Thank you.
Thank you so much, Dr. Lozcano. That was a very insightful presentation. Exciting to hear of some of the other research work that's happening in our region and down further south. So we are going to shift over to the question and answer. I'm going to pass over the baton to my colleague, Keith Abels, who's going to run the questions. And so please keep entering questions into the Q&A window if you'd like to have a question answered by one of our speakers. And that includes Dr. Lozcano and also Sean and Julian from Jackson Family and Dr. Beniston, if you have some remaining questions about his talk. And before we dive into that, I wanted to do one little poll in terms of wanting to see what folks would be interested in hearing about at a future workshop. So hopefully you can see there's a little poll that should have popped up in your window asking what soil health related topic would you like to see at a future workshop? And you should be able to just press a button and vote on you should be able to select more than one topic. So I'm going to give a minute or two for folks to enter their choices. And I see some votes coming in. Great. So again, this is, we are listening to you. And so please, the more feedback we get from you, the more we could frame our future events and future grant funding projects tailored to what you folks are interested in learning more about and getting insights for your own management decisions. All right, one more second, last chance to vote. And all right. And I'm going to end the poll. Looks like we have a tie for soil microbes and tillage and carbon sequestration. Oh, and cover cropping for soil health. So a couple of different topics that we could look into doing some cool future workshops for. All right, well, Keith, I hope you are ready to take it off with the questions. I see we have a couple in the Q&A window, and I think a few folks might have added some in the chat window. So I will be passing it to you to start with the questions.
Okay, I'm ready. Can you hear me?
Yes.
Okay, hello everybody. This is Keith Abels and I'm the Soil and Water Specialist at the Sonoma RCD. So I got some questions. Feel free to keep adding to the question list. I'm going to ask a couple for Cristina first, just because you just finished your show, your presentation, and they're related to that. One of them is at the J-Lore trial, what carbon to nitrogen ratio compost is being used, and then if you know, and another related question is in the Edna Valley vineyard, were chemical herbicides used in that vineyard, and could that be an element of the variation from tractor to vine row?
All right, so in terms of the carbon, the C2N ratio, the compost we're using is a mix of manure and garden waste. It's a compost that is produced by Cal Poly. And I think, off the top of my head, I think it's 11 or 12 CN ratio, but I need to double check that. Initially, we wanted to use grape pomace based manure, based compost, but we didn't find a lot of grape pomace compost being available. So we ended up using more of a mixed manure and green waste. And then the second question was in terms of that Edna Valley trial and whether we used herbicides. We did not, there was no herbicide being applied under the vine. The weeds were managed by cultivation. So it was more mechanical weeding, not chemical. So yeah, there's a series of management differences between tractor rows and vines, right? Not just removing the weeds, but also water, irrigation water is applied under the vine, fertilizers are applied under the vine, and all that has an effect on the food web. And so I didn't show it here in this presentation, but we actually looked at how fertilizers affect the food web under the vine. We compared synthetic sources of nitrogen to organic, so compost. And we've seen there was a very strong difference. So applying organic fertilizers actually decrease the amount of plant parasitic nematodes under the vine. This is a paper that it's under review that we hope to be publishing pretty soon. But it's really exciting to see these very strong impacts to the food web. Yeah.
Okay, thank you. Thank you. The next question is, and is one that I might be able to answer, but someone asked about how long the study will be continuing. And I can tell you that we plan to go out to continue the treatments and go out in next year and do soil sampling again. That'll be 2021. And then in 2023, we plan to do our final set of soil sampling unless we decide to extend the trial. But if Anya or anyone from Jackson Family has anything to add from that, I just wanted to clarify that. And then another question that came in is on compost, and you've been applying in spring. This is really, I think, a question for Sean. Why do you apply for spring? I know you answered this offline, but if you could answer for the group, like sort of what is your preferred time to apply compost? And also related, what is your preferred frequency? I know that this grant requires us to do it yearly, but if you had your choice, how often do you want to be applying compost?
Yeah, so with the timing of the application, it's always post-harvest for us. But really it's the rain that determines, when we're getting into the vineyard from post-harvest all the way through bud break. And, a lot of the vineyards in the Russian River that I farm are on valley floor where, it can get pretty wet and get tractors stuck. So, sometimes we're able to get into the hillside vineyards that dry out a little faster. But really what I'm doing is is a 3-ton compost application every other year, and that's for all of the vineyards I farm in Anderson Valley, Sonoma Coast, and Russian River.
Okay, thank you. The next question I have could be for Cristina or Josh, and the question is, what effects do you anticipate long-term for building soil carbon reserves below the 6-inch level using these healthy soil practices? And do you think they can be achieved even if a vine row is tilled at a depth of 6 inches every other year?
Yeah, I guess I'll take a first stab at this, and maybe Cristina, you can follow up as well. You know, there are a handful of studies that I'm aware of that have documented that things like compost application are resulting in changes in soil carbon deeper below the surface of the soil. The recent study that I mentioned in my talk, that's part of the deal there, is they did a real deep soil sampling, and they said that that gave them a different outcome than if they had just sampled in the surface. I mean, one of the challenges there is just the cost involved. It takes a lot more time, and it's a lot more samples for measuring deep in the soil. I suspect that the treatments of compost application and cover cropping and reducing tillage will lead to longer term impacts deeper in the soil. The second part of the question, I think it's harder to predict what happens deeper in the soil when you are doing regular cultivation. There's definitely a lot of older soil research that shows that surface cultivations have negative impacts on carbon levels below, deeper in the soil. There's a term called the priming effect, where when you make carbon decompose somewhere in the soil, it can trigger more decomposition below. So it's harder to predict. So my response to the first part of the question is yes, I think these practices would lead to changes deeper down. I think the second part is it's hard to answer what the effect of tillage would be lower in the soil, but I suspect it may not be good if it were not coupled with these other, I think compost is the key, right? The piece that I showed from Davis showed that with the addition of compost, they were still tilling those fields every year, but they were able to sequester carbon, and they were applying the same that we did in the trial, 5 tons per acre, which a lot of farmers think is a lot. But I think if you want to till the soil and you want to sequester carbon, you got to put a lot of compost into it, would be my response.
Yeah, I don't have much to add to that. I think there's definitely a great potential for improving carbon at depth. And, as you just said, there's studies that show that by managing soils using compost or cover crops, we are actually, yes, effectively increasing carbon at depth. And then that could actually be substantial in terms of the big numbers of carbon sequestration. And that should be something we should explore, definitely.
Great, thank you. I have a question. I think this is perhaps for Josh, which is when you're applying the compost, but then you had your carbon content differences that you noted after 2 years. How do you subtract, or how do you account for the additional carbon that just comes from the compost itself versus the overall activity it stimulates by applying the carbon beyond just the compost itself?
Okay, so I'll try to answer this one, and maybe get Cristina to comment as well. But I guess I would start to address that question by saying that when the term soil compost and the term soil organic matter are pretty broad in terms of what kind of actual material we're talking about, right? And then soil carbon really encompasses everything from freshly killed, freshly dead plant materials in the surface of the soil to tiny little molecules of carbon compounds that have been secreted by roots or that are microbial byproducts. And so with compost, I think part of the reason that compost is you know, maybe the fastest solution for building soil carbon is that it essentially is sort of instant soil carbon. All of the pieces and particles that are below a certain size threshold really, in my mind, fall under the definition of soil carbon. We try to remove live plant materials from our samples. But so no, there's no real subtraction. And I think, you know, that's part of why it goes up, is because you're adding, with compost, you're adding plant residues that have already been decomposed to a certain extent into the soil. So you're adding material that is essentially the same as soil carbon, and that is soil carbon below a certain size. And so that's why it goes up faster with compost application than something like tillage, where you're not adding a new material that is carbon. So that's my response there.
Thank you. Cristina, do you have anything to add?
Yeah, so if I understand the question right, is that how much is compost contributing to that increase in carbon? So my experience is that what you apply in compost to the soil is really a very small amount of carbon as compared to what's already in the soil. And so what you're doing, the contribution of compost, rather than being a direct addition to soil carbon, which it is, but the most important effect of compost, it's the stimulation of microbial activity and microbial growth. And also the trickle-down effects on the food web that also end up increasing the cover crop growth. And all that, it's kind of a synergistic effect that adds more carbon to your soil at the end of the day. Because the cover crop growing more, it means that it's going to sequester more CO2 from the atmosphere and put it into the soil through the roots, right? And if the cover crop is then terminated and incorporated into the soil, then that's even more carbon. So a little bit of compost goes a long way because of all the ecological effects that it has in the soil. So it's actually a synergy in the carbon cycle.
Excellent. Anya, I'm not sure I got your question about that you could add to after Cristina about when we sample before composting or after.
Yeah, I just wanted to clarify, I didn't catch if Josh mentioned this, but the annual soil sampling we do at the demo trial at Saralee is we do the sampling ahead of compost application. So we're not grabbing any of that compost that is applied that year as part of the sampling. So it's whatever was applied the previous year has had a year to get incorporated and start working through the soil profile.
Thank you. The next question, I'm combining two questions here. I think Sean might take, we'd like to ask you this is, what do you see as the impact of standard vineyard herbicide use under the vine on microbial soil health? And how do the costs compare of moving from herbicide use to mechanical management under the vine?
Okay, so I can't really give any definitive answer on what the microbial soil health would be or the difference. We are doing the soil DNA tests, which are pretty interesting. It's just another data point, right? But we're looking at what kind of organisms were living in the soil, when we were using pre-emergent herbicides, and then post elimination of herbicides, we're looking at what the soil DNA is. every year. So we're keeping track of that. I'm not sure if it's a legitimate data point or not, but we're trying to collect as much data as we possibly can to see if we're seeing any benefits with regard to like these beneficial organisms that are in the soil. And then with regard to cost, yeah, that's farming 2,500 acres. Like I couldn't just convert everything over to no herbicide. I'm still using some organic herbicides here and there. You probably know Weed Slayer or some of the others, but I've eliminated, of course, Roundup and all the pre-emergent herbicides. But yeah, it took time because I had to put in the Clemens implement, the the Pelonc sunflower. We're trying out all these different weeders, mowers, and crimpers. And so these are all things that I had to get into CapEx budget every year. And I took it slow. I didn't take it all on at once because it's also labor too. It takes more time to get through a field with the weeder than it is than the spray rig. But yeah, now that we have everything working smoothly, we have some redundancies. We have 5 Clemens machines and we got 3 sunflower machines. So it's I think the initial costs are there, with your implement and people learning how to use the equipment. Of course, in a place like Saralee, it's so much easier to use the Clemens. It's flat. The soil is a sandy loam, so it's really easy to work the soil. But as you get into the hills, where you have some terracing and the ground is a little bit harder, your timing is more crucial in the hills because at some point it might be too hard to cut the soil with the implement. So yeah, I mean I don't see too big of a cost difference now that we're getting good at it.
Thank you, Sean. That was great. I think we have time for one more question. And a question came in, and I think Cristina might be the best person for this, have you looked at the impact of animal grazing on soil organic matter? Can you touch a little bit more on that, please?
Yes, so we are looking at that in our trial. And so there's different ways of looking at soil organic matter, different fractions of soil organic matter. We are looking at one hand total carbon, that it's an indicator of how much organic matter is in the soil, but also at active carbon, which is the carbon that it's used by microbes, and therefore will have like a more impact in the soil health and soil fertility in general. And so we didn't see differences in the total carbon of the soil. So there was not a total change in net organic matter, but we did see that at the 6 to 12 inches depth of the soil, there was, right after grazing, an increase in active carbon. And so this is something that we think is happening because after the sheep is grazing and in the plants, there is like a release of root exudates. There's a release of carbon from the roots that might actually feed the microbes and constitute like a feedback. So the microorganisms release nutrients that allow the crop, the cover crop, to grow back and grow back stronger. So yeah, that's the only effect we've seen. And right now we're looking more in detail at exactly what are the microbial processes that are happening right after grazing. But I hope to have more results by the end of the year. So far, we're still analyzing samples.
Great, thank you. I wish we had time for more questions, but I see we've reached just about 11 o'clock. So I'm going to turn it back over to Anya, who has some parting comments. Thank you.
Alrighty, thank you so much. And I know there might have been a few questions that we weren't able to get to. And so I, first of all, just want to say thank you for all of you taking time to join us today and tuning in. We did record this webinar, and so within a week's time or so, we will be sending out the link to everyone who registered. So everyone who participated today and those who just signed up and weren't able to tune in. If you have friends you think would find this presentation insightful, please feel free to forward that, that video link. And I'm also adding some information into the chat window. And so if you're interested in learning more about some future workshops and webinars that we'll be hosting, you're welcome to sign up for the RCD newsletter. That's one way to keep in touch on future events. The second one is keeping tabs on the Soil Hub. So there's a regional effort that is being shared across several different resource conservation districts and partners like NRCS and UC Davis, and also a suite of other, and the junior college and a bunch of other researchers and local organizations that have come together to try to develop these kind of soil health parameters and a better understanding of what good soil health means for the North Coast region. And so soilhub.org is the best way to pay attention to that effort and also hear about events that the different RCDs will be putting on for education, and also opportunities to participate if you'd be interested in getting to know the soil health on your particular property. And I also included the emails for Dr. Beniston and Dr. Lozcano in case you did have a remaining pressing question you wanted to direct in their way. And again, thank you so much to all the panelists who presented today. We're very excited about this partnership, and look forward to next year's data and continuing on with learning more about soil health and being able to share that with everyone else, so we can all strive to do some more exciting stuff on our properties and keep the soil healthy for the rest of us and future generations. So with that, I am going to say farewell and close out the webinar, and thank you all and have a great rest of your day.
Thank you.
Thanks.